Welcome to Pharmaceutical and Medicinal Chemistry. Module 1 in this course is Introduction to Pharmaceutical Chemistry. This module is subdivided into three chapters. Chapter 1.1 is Terminology, Drugs, and Receptors. The instructor for this chapter is Dr. P. Sharada, Selection Grade Lecturer in Chemistry, St. Joseph's College for Women, Autonomous, Vishakhapatnam. Learning Outcomes. By the end of this chapter, the learner would be well versed with the basic scientific terminology in pharmaceutical and medicinal chemistry. Learner would also be able to distinguish an ideal drug from others, comprehend the mechanism of drug receptor interaction, and also differentiate and categorize receptors based on drug receptor interaction. Contents: Introduction to Pharmaceutical and Medicinal Chemistry, Terminology, drug definition and characteristics, receptors, mechanism of drug receptor interaction, and types of receptors along with their mode of action. What is pharmaceutical and medicinal chemistry? Pharmaceutical and medicinal chemistry lies at the interface of pharmacy and chemistry. While pharmacy is the scientific study of discovery, synthesis, dispensing, review, safety, and affordability of medicines, chemistry is the scientific study of matter. It also includes studies on structure, composition, synthesis, properties, and changes in composition and properties due to chemical reactions. Pharmaceutical and medicinal chemistry is an interdisciplinary science which involves the design, development, synthesis, and bioassay of biologically active chemical entities which can be used as medicines or drugs. It also deals with ensuring and assuring their quality as well as safety. This multidisciplinary science includes concepts and applications of organic chemistry, physical chemistry, biochemistry, computational chemistry, molecular biology, and biostatistics. Terminology. What is pharmacy? Pharmacy is also called as pharmaceutics. It is simply the science of drugs. It deals with the preparation, properties, analysis, standardization, and dispensing of drugs. It also includes studies on drug delivery system and pharmacodynamics and pharmacokinetics of synthetic drugs or medicines. Pharmacology. What is pharmacology? Pharmacology is a branch of medicine, biology, and pharmacy. It is the study of how drugs interact with processes of living systems. It includes studies on properties, mechanism of biochemical action, physiological effect, therapeutic use, and also adverse effects of drugs. Pharmacophore. Pharmacophore is a part of the drug. According to IUPAC definition, pharmacophore is an ensemble of steric and electronic features that is necessary to ensure the optimal supramolecular interactions with a specific biological target and to trigger or block its biological response. Simply, a pharmacophore is the part of the drug with a specific steric and electronic arrangement which is necessary to either trigger or block a response in a living system. Pharmacophore is a three-dimensional arrangement of atoms and groups. It interacts with the receptor through the pharmacophoric points and it is responsible for the biochemical effect generated in the system. The common pharmacophoric features are hydrophobic centroids, aromatic rings, hydrogen bonding functional groups, cations, and anions. Through these pharmacophoric points, the pharmacophore produces the desired biochemical effect. Similar pharmacophores produce similar effects. As we all know, ibuprofen, ketoprofen, naproxen, all these are anti-inflammatory drugs. If we have a closer look at their molecular structure, we can see that aryl methyl acetic acid is the common pharmacophore in all these drugs. So, this pharmacophore generates the anti-inflammatory effect in the system. Similar pharmacophores produce similar effects. What is pharmacopoeia? Pharmacopoeia is an official list of recognized medicines. It also includes the effects as well as usage directions for the medicines that are listed under pharmacopoeia. What are drugs? Drugs are the natural or synthetic chemical messengers which interact with macromolecular targets in living systems and produce the desired physiological or psychological change. They are molecules of low molecular mass which are used in the diagnosis, cure, mitigation, treatment, and also prevention of disease. What are the characteristics of an ideal drug? Drugs should be non toxic, biocompatible, and biodegradable. They should be physico chemically stable, restricted but predictable action should be there without any side effects, controllable rate of relief, and uniform capillary distribution, easily eliminated through normal metabolic pathways without any accumulation in the system. And drug carriers should be biodegradable, drug delivery system should be simple and reproducible. There should not be any leakage of drugs during transit. So these are the characteristics of an ideal drug. It should be non-toxic, biocompatible as well as biodegradable. It should be stable. Its action should be restricted only at the target site. The rate of release of drug should be predictable. It should not cause any side effects. And there should be uniform capillary distribution of the drug at the target site. Drugs should be easily eliminated after their action. They should not get accumulated in the system. And also, drug carriers should be biodegradable. What are receptors? Receptors are macromolecular proteins which are present either on the cell surface or the inside of a cell. They are the receivers of messages carried by the drugs or the naturally existing ligands. They are the receivers of chemical messages which are highly specific. They can only bind to specific ligands or drugs. Receptors bind with the drugs or ligands through the receptor sites. Receptor sites have a complementary stereochemical structure matching with that of the natural ligand or the synthetic drug which is meant to bind with the specific receptor. Drug and receptor interact through a minimum 3-point attachment. The mechanism of drug receptor interaction is more like the lock and key. Both the lock as well as the key are highly specific to one another and their interaction is also temporary. Once the lock is open, there ends the interaction. The effectivity of drug receptor interaction is controlled by selectivity, affinity, and efficacy. Selectivity refers to the mutual preference between the drug and the receptor. Affinity is the inclination to get bound to one another, and efficacy is the ability to generate the desired response. So, the effectivity of drug receptor interaction is governed by three factors: how the drug prefers a particular receptor, or conversely, how the receptor prefers a particular drug, or what is the inclination between the two to get bound to one another. Once they are bound, what is the ability of the drug receptor complex to generate the desired response. The binding and generation of response takes place in three stages. First, the receptor recognizes the drug or the ligand. Next, the receptor binds to the drug, or there is binding between the drug and the receptor through hydrogen bonding or ionic interactions or van der Waals forces, and this binding is followed by translation of the signal into effect. So the mechanism of drug receptor interaction involves recognition, binding, and translation. Finally, the desired response is generated. The interaction between the drug and the receptor is highly specific. We can see here the lock is violet in color, the key is violet as well as gray, but the pharmacophoric points on the violet key are matching with that of the receptor, are complementary to those of the receptor. So it is only the A drug which binds to the receptor and generates the action. Though B is available in the medium, it cannot bind to the receptor because it doesn't have the complementary stereochemical features. Types of receptors. Receptors are broadly classified into 5 categories. The first among them is intracellular receptors or cytoplasmic receptors. Cytoplasmic receptors, as the name itself indicates, they are located in the cytoplasm. So, only those drugs that can travel through the cell membrane or the plasma membrane can interact with these receptors. So the drugs have to be hydrophobic, and they should traverse the plasma membrane and get bound to the receptor site that is present on the cytoplasmic receptor. Once the drug binds to the receptor site, the receptor changes its shape and exposes the DNA binding site present on it. You can see here the signaling molecule is red. It is hydrophobic. It is traveling through the cell membrane. After traveling through the cell membrane, it gets bound to the blue receptor in the cytoplasm. Once it is bound to the blue receptor, the one particular site that is called as the DNA binding site gets exposed on the receptor. Now, this receptor travels into the nucleus along with the drug, and then it binds to the DNA. Once it binds to the DNA at a specific site, it triggers mRNA production inside the nucleus. So, this particular receptor does not involve other receptors in translating the signal carried by the drug. We can see very clearly the drug molecule is red. It is outside the cell. Next, it traveled through the cell membrane because it is hydrophobic. It got bound to the receptor, which is blue in color. Once it got bound to the receptor changed its shape and it exposed the DNA binding site. Then both of them together travel through the nuclear membrane, enter the nucleus, get bound to the DNA, and then the RNA synthesis is triggered. The second type of receptors are cell surface receptors or transmembrane receptors. As the name itself indicates, these receptors are present on the cell surface. These receptors consist of three parts: the exterior drug binding part, the long hydrophobic cell membrane spanning part, and the intracellular part. You can see here the blue block is the receptor. The V cut in the block is the binding part, which is present exterior to the cell membrane. The long blue part is the cell membrane spanning hydrophobic part, and the inside of the receptor is the part that is present beneath the cell membrane. Once the external, now our drug is triangular in shape, matching with the slot on the receptor. This triangle gets bound to the receptor. We can see here drug receptor complex is formed. Signal transduction takes place once the receptor binds to the drug at the receptor site, and external signal is converted into internal signal, and response is generated by the intracellular part of the receptor. The most salient feature of this receptor is that this receptor can generate the desired response without taking the drug into the cell. That is to say, the drug remains outside the cell, but still the effect or the signal that is carried by the drug is translated and generated in the form of a response inside the cell by this type of receptors called as transmembrane receptors. The third type of receptors are ion channel linked receptors. You can see these receptors are green in color. We can see that they are also present on the cell surface, but in the middle of the green receptor, it is closed. But once this yellow color ball is the drug or the signal carrying ligand. This signal carrying ligand binds to this green color receptor at the specific site, and we can see in the second part of the diagram that the channel is open now. Once the drug binds to the receptor, the channel opens up. Once this channel opens up, ions of specific charge and dimension can travel through the channel and enter the cell. Otherwise, these violet dots or violet circles, they are present in the extracellular fluid. That is to say, they are present outside the cell. But once this yellow ball, which is our signaling drug or ligand, binds to the specific receptor site, the channel opens up and we can see that the violet balls are traveling inside to the cytoplasm. That means the ions which are present outside, they travel into the cell through the channel, but not all ions can travel. Only those ions with a specific dimension and charge can travel through the channel into the cytoplasm. Now, these receptor proteins are present on cell surface. Receptor site binds the drug. Protein undergoes conformational change. That means change in the shape of the receptor takes place. Conformational change means a three-dimensional change in the structure takes place and opens an ion channel across the membrane. Ions of particular size and charge pass through the membrane into the cell. The fourth type of receptors are G protein coupled receptors. These G protein coupled receptor binds to the specific binding site on the receptor but remains inactive. Mind you, G protein is present in the cell and the receptor also is in the cell. G protein binds to the receptor, but nothing happens. Both of them together will remain inactive. But once an inactive GPCR stands for G protein coupled receptor, which is inactive. Once this inactive GPCR binds the drug, once it binds the drug, it becomes active and it undergoes some changes in its shape and it splits into two subunits. One is alpha and another unit is beta gamma. So you get two subunits. Sometimes both the subunits are active. Sometimes one subunit is active and the other one is inactive. These activated fragments activate other proteins and generate the desired response. In the whole process, there is an exchange between GTP and GDP. Later, GDP and GTP, again the hydrolysis of GTP takes place so as to produce GDP. The last type of receptors are enzyme linked receptors. These receptors are also present on the cell surface. The intracellular regions of these receptors, either they themselves are enzymes or they can activate an enzyme. So we can say that enzyme linked receptors are in three parts. One part is the drug binding part. The second part is spanning the cell membrane. And the third part is present in the cytoplasm. The part that is present in the cytoplasm can itself act as an enzyme or it can activate an enzyme once The yellow color drug molecule binds to its receptor site. The intracellular part itself can also be an enzyme. Drug binds to the receptor site, enzyme gets activated, and we can see here the substrate plus enzyme will produce the desired effect inside the cytoplasm. So, what have we learned in a nutshell? We have seen the introductory part of pharmaceutical and medicinal chemistry, what the terminology, pharmacophore, pharmacopoeia, pharmacology. Then we have seen what are drugs, what are the characteristics of an ideal drug. Then we have learnt about the mechanism of drug receptor interaction. Prior to that, we also learned something about what receptors are, what is the mechanism of their interaction. Then based on the interaction between the drug and the receptor, how many different types of receptors can be identified, all that we have learned in this chapter. So in a nutshell, we have learned about the terms pharmacy, pharmacology, pharmacophore, pharmacopoeia. Then we have seen what drugs are. Drugs are chemical messengers required to generate the desired biological response. Then we have seen what receptors are. They are specific drug binding macromolecules present on or inside the cell surface. And what is the type of drug receptor interaction, which is highly specific, more like a lock and key. Then there are five types of receptors, and different types of receptors generate different types of responses after binding to the drug. Thank you.